Hi, today I'm gonna introduce an article named In Vivo Labeling by CD73 Marks Multipotent Stromal Cells and Highlights Endothelial Heterogeneity in Bone Marrow Niche. This article provides a genetic model enabling the identification and imaging of multipotent stromal cells in vivo. In the bone marrow, there are at least two major types of endothelium have been identified: the sinusoidal and the arteriolar endothelium. And near to this endothelium, there are a very important cell called multipotent stromal cells, or MSCs, can always be detected in perivascular location in various organs. And all these cells with hematopoietic stem cells together construct a hematopoietic stem cell niche in bone marrow. The MSC isolate from bone marrow is a colony-forming fibroblastic cells, which have stem cell renewal capacity and differentiation potential. It can differentiate into various mesenchymal cells like osteocytes, adipocytes, and chondrocytes. The MSC from bone marrow can be isolated and expanded in vitro, and there are three ways to characterize the MSCs. We can perform a CFU-FS in bone marrow bug culture. CFU-FS is a well-established method for the quantification of MSC from human bone marrow or other tissues. The clones of CFU-FS represent stem cells and progenitor cells with no hematopoietic differentiation potential. So we can characterize MSC by his fibroblast-like morphology, or we can examine the differentiation potential or analyze the markers. To identify the MSCs in vitro, but in vivo, we don't have a specific marker for isolation and identification in vivo for MSCs. So for checking the MSC in vivo, they generate a CD73 EGFP reporter mouse. So they constructed a BAC vector. BAC is a bacterial artificial chromosome vector which contains the EGFP and the CD73 genes. And this vector was transferred into MSC, and the selected MSC can express the CD73 and EGFP. And this MSC cells was confirmed can be differentiated, but the EGFP fluorescence were lost during the differentiation. So this indicates that CD73 is a suitable marker for the undifferentiated multipotent cells. And then they find out the EGFP expression in kidney and lung, which have a higher mRNA levels for of CD73 in epithelium. These results confirm the specificity of the transgene, but whether these reported mouse can be used for the isolation of MSCs from the bone marrow in vivo is still unclear. So to confirm the function of transgene mouse that enable the detection of MSC, we want to know is that this reported mouse can detect the MSC in various organs and in bone compartment. So they perform an immunostaining in uterus, liver, and white fat, which usually used for the MSC isolation in vitro, and they found that the EGFP expression in these three organs and these EGFP positive cells are near to the vessel which marked by Pecan, the endothelium marker. And then to characterize the nature of EGFP positive cells, they performed a flow cytometry, and they found that this EGFP positive cell express the mesenchymal marker CD146 and SCOR1, which is the typical marker of the MSCs. And they are all CD45 negative, which is present in all differentiated hematopoietic cells, except the SR cells and plasma cells. And then they examined the EGFP positive cells from white fat, which is the most frequently used for MSC isolation in vitro beside the bone marrow. They found that these EGFP positive cells are lack of hematopoietic markers like CD45 and CD11B and express the typical MSC markers CD44 and SCAR1. What's more, they found that these EGFP positive cells can differentiate into osteogenic, adipogenic, and chondrogenic lineages. Taken together, this research shows that the EGFP positive cells of white fat are MSC cells because they show the typical fibroblastic morphology and they express the mesenchymal cell markers and they can differentiate into three lineages. So after knowing the MSC in various organs can be detected by these transgenic mouse, we want to know next is this EGFP can label the MSC in bone compartment or not. So they analyzed the EGFP expression in developing bone of embryo and in the growth part of elder long bones. And they found that in rate of E16.5 embryo, the EGFP could label the perichondrion, which contain the mesenchymal precursor cells. And in elder femur epiphyses, they also found the EGFP can label the chondrogenic progenitor and the early chondrocyte, and the florism will be lost after the differentiation. This result shows that the EGFP positive cells in this bone compartment could be MSCs. In addition, in time-lapse microscopy, they found that the EGFP positive cells can propagate and display the MSC morphology. And then, as no MSC reside at the inner endosteal surface of cortical bones, they screened the diaphysis section by microscopy. And then they found some rare EGFP positive cells were lining the bone, and these cells were mesenchymal marker positive and endothelial marker negative cells. So they want to know what is these EGFP positive cells. Are they MSCs? Distribution of these cells in bone. They performed the flow cytometry on freshly isolated CD73 EGFP mouse bone marrow cells. And they found that only very few percentage of these cells were CD45 negative and EGFP positive cells. So they did another flow cytometry to reconfirm the MSC nature, and they found that there are almost 70% of these EGFP positive cells are SCAR1 positive. So after confirming the MSC nature by the markers analyzed, they want to confirm the economic capacity and the differentiation potential of these EGFP positive cells. So they plotted whole bone marrow cells to perform CFU-LFS assay, and after 2 weeks, they screened the cultures for the EGFP expression in developing colonies. I picked a single colony to do the differentiation assay, and they found that all the colony of CFU-LFS contained the EGFP positive cells, which confirmed the EGFP cells have the typical MSC-like morphology. And as shown in Figure E, there are only almost 30% of EGFP positive cells in this bug culture. This represents a heterogeneous population of cells. There may be some CD45 positive blood cells. And after 25 days, the EGFP positive cells can differentiate. Figure G, there are some EGFP florism loss during the differentiation. Is there only 30% of EGFP positive cells in this culture? They saw the EGFP positive and negative MSC-like cells from this culture and to see the MSC feature. And as we can see in the Figure B, for the EGFP positive or negative cell, they have similar markers expression levels. But what is different is the EGFP positive cells can differentiate, but the negative cells no, which indicate the CD73 molecule is very important for the multipotency of these MSC cells. And then they took their CD45 negative and EGFP positive cells to do the flow cytometry again to sort these cells and to culture in our CFUF again to see is there's a proliferation of these colonies, and as shown in this figure, all these colonies are expressed EGFPs, and these cells can also give rise to colonies expand by past aging, which show the cell renewal potential of these cells, showed by Fig E, enhanced or GFP frozen in the colony. So, in conclusion, these cells from bone compartment or MSC can be identified and isolated by this reported mouse. But we still don't know the distribution of these cells in bone. So by immunostaining and confocal and two-photo microscopy, we can see that these EGFP positive cells have no hematopoietic nature and have the sinusoidal structures. And interestingly, these EGFP positive cells also express their endothelial markers, and they are near to the mesenchymal perivascular cells. So they want to know these elongate EGFP positive cells were sinusoidal endothelial cells or not. So they culture these cells in their mesenchymal medium, and after 1 week, we can see there are no hematopoietic markers in these EGFP positive cells. But after 3 weeks, there are significant endothelial markers in these overlap these EGFP positive cells. So they want to find out the drainage of these cells. So they did the flow cytometry and the RNA sequence, and they divide into 3 populations, which the double positive express sinusoidal strains and endothelial strains may be indicated as SECs, and for the EGFP single positive express mesenchymal strains indicates MSC cells, and for the pecan positive single positive, it may indicate the AEC cell. And the further RNA sequence analysis shows that the sinusoidal cells express higher level of the mesenchymal strains than the arterial endothelium cells, as well as the E-selectin and VCAM1 genes, which can use to distinguish these 2 endothelium cells. And they also performed a gene set enrichment analysis, which they found that the sinusoidal cells can express more or higher stromal stem cells than the arterial cells, which are indicated endothelial heterogeneity in the bone marrow. So in conclusion, this CD73 EGFP reported mouse can detect MSC in uterus, liver, white fat, and the bone compartment. According to its MSC-like morphology and differentiation potential and cell renewal capacity, we can confirm these EGFP positive cells are MSCs, and these reported mouse can identify and isolate MSC from the bone compartment in vivo. And moreover, by analyzing the distribution of the EGFP positive cell in bone compartment, they confirm that this CD73 EGFP can specifically label the sinusoidal endothelial cells. And the RNA sequence analysis shows that different endothelial cells have different gene expression in different cell population, which reveal the endothelial heterogeneity in HSC niche. For discussion, could CD73 EGFP still label the sinusoidal endothelial cells in the cancer metastasis process like EMT and MET, the transformation of the endothelial cells and mesenchymal cells? And what is the relation between the MSC and the type H endothelium, the specific type of the EC cells in the metaphysics.